it's my great honor actually to be invited to join this webinar to share some of our recent work on the topic of biofilm engineering. But today I will specifically talk about the biofilm lifestyle of common monomers in the environmental context. So my group in South Sea focuses on the understanding the biofilm lifestyle of environmental bacteria and try to apply the insights from the biofilm contaminant interaction to improve or develop the biofilm biotechnologies. So specifically, my group, they focus on the understanding the interaction, understanding the biofilm lifestyle and developing tools to study biofilms. And we are also interested in engineering the beneficial biofilms so that it can be used for bioremediation or the resource recovery. It can also be used for chemical synthesis. And we are also interested in controlling the detrimental biofilms in the environmental context. For example, the water purification membranes. So in this presentation specifically, I will use the example of the biofilm contaminant interaction and how we can use the insights to improve the performance of the biofilm-based bioprocess. And in this specific work,
Ben,
yes.
Sorry to interrupt. I think you haven't shared your screen.
I'm sharing that. Oh,
okay. Is everyone able to see
can
the
I see
screen?
that?
Mark?
No, we can see you, Ben, but not the slides
No.
at the moment.
Oh,
Yeah.
We can't. Yeah.
maybe because you shared it and then it stopped my sharing.
Okay. Yeah.
How
Now
about
it's
this?
coming up.
Yeah.
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 That looks good.
Oh, that's bad, huh? Okay, never mind. So the yes. So this is basically I just mentioned the my key research areas, and I'm going to share some of our recent work on the biofilm contaminant interaction and how we can use insights to make it to make the biofilm-based process better. So we used common monomers as a specific model organism in this study. And why common monomers? It's at the time when we decided which organism to use, we actually look at the relevance of the organism in the wastewater treatment plants. And if you can look at this map, so the numbers here are the relative abundance of the common monomers, this group of organisms in the wastewater microbiome. So in different areas in the world, and you can see the percentage is very high, considering the very high diversity in the wastewater microbiome. So 1% or 2% is really extremely high already. And the interesting thing is, this group of organisms are also reported as the primary surface colonizers in the membrane bioreactors for domestic water treatment. So this is a very interesting group of organisms, and we still do not know much why they thrive in this environment and why and what exactly they are doing there and why they can be primary colonizers. So that's why we use one common monomers bacteria, that's common monomers testosterone, as a model organism, and to look at a very simple interaction. That's the bacteria interact with a specific contaminant, and in this case, the contaminant is 3-chloroaniline or 3CA, and this bacteria is known to be a aerobic bacteria. So so many researchers they know about this bacteria, so they will wonder, it is very clear that the interaction between this bacteria and 3CA is very simple. In the aerobic condition, this bacteria is able to completely degrading 3CA to carbon dioxide and water. So that's it. Yes, that's just one aspect of this interaction. Now, the question that we want to address is actually, what if we introduce this contaminant to the biofilm of this bacteria, and then what happens? So this is the simple experiment. We grew the biofilm for 4 days, and then after that, we introduced 3CA at a concentration of 200 milligram per liter. So that's a quite low concentration, and at this concentration, the bacterial viability is not affected at all, and in fact, the bacteria are able to degrade 3CA at this concentration. But the interesting thing is that if you look at this, the biofilms start to disperse once we introduce 3CA. And we quantified the number of cells move away from the biofilm at a unit of time. And that's what we call the detachment rate. You can clearly see that after we introduce 3CA, the rate of detachment significantly increased and the biovolume reduced. And that's because of the dispersal of the biofilm. Now, the question here is what happened? So then we, in a transcriptomic analysis, and we observed a list of genes, downregulated genes, and many of them, they are involved in CDJP synthase. So to biofilm researchers, I believe most of you are very familiar with CDJP. And basically it's a signaling molecule. And when the CDJP level is low and the biofilm information will be reduced, or the biofilm will be the detachment will be increased. So in this case, we have a lower expression of the CDJP synthesis, that's the DGC activity, and then we quantified the CDJP level, and actually, indeed, we have a lower CDJP level in this case. Now, as an engineer, I always wanted to ask the questions. Actually, now you introduce 3CA or biofilm disperse, but what if we want to use the biofilm to degrade 3CA? Of course, we want the biofilm to stay longer. So how can we make the biofilm stay longer? Is it possible that we push up the CDJP level and we can keep the biofilm stay longer? So a very simple experiment, we introduce a CDJP synthase from E. coli into this Commomonas testosterone, and then we saw the increase of CDJP level, and we also saw the higher biofilm formation capability. Now, we grow this bacteria in a packed bed reactor, and then we introduce 3CA, and we quantify the cell detachment from this packed bed biofilm reactor. And this is what we see. For the wild type, once you introduce 3CA, you see the detachment rate substantially increased. But for the transconjugant, so that's the one that actually with a higher CDJP level, you do not see much difference, even if you introduce 3CA to them. So that means if we increase the CDJP level, we are able to keep the biofilm stay longer. So that's kind of interesting to us because I'm interested in beneficial biofilms for the many different applications, including biochemical synthesis, for example. Now, this suggests that we are able to improve biofilm robustness by manipulating the CDJP level. But the problem here is actually the CDJP level is reduced by 3CA, and then the CDJP level is increased by introducing a CDJP synthase gene, and we constitutively expressed it. Now, along this line, we were thinking, can we control the biofilm dynamics through controlling the CDJP dynamics? And along this line, we actually designed the biofilm that are able to be controlled by using light. So in this specific design, we have the light responsive DGC and light responsive the PDE. So when we have the biofilms that are exposed to the near infrared light, and CDJP will be synthesized and the concentration will be increased. When we have the blue light, CDJP level will be reduced. So in this case, if we have a biofilm here, we introduce NRR light and the biofilm becomes thicker. And then if we change to the blue light, the biofilm becomes thinner. So that's because of the CDJP dynamics, and the biofilm formation dynamics is also response. So that this is kind of the very simple idea. Now, we have also demonstrated the applications in the biochemical synthesis, biocatalysis. That's actually the controllable catalytic biofilms for the chemical synthesis, and quorum quenching controllable biofilms with the quorum quenching activities for biofouling control. We also demonstrate the application of a controllable electrochemically active biofilm for the bioelectrochemical applications. So that's based on the idea of using the CDJP dynamics to control the biofilm dynamics. Now let's come back to the common monomers. So we talked about common monomers, and they are able to once we introduce 3CA, the biofilm will disperse. But if we increase the CDJP level, we can keep the biofilm stay longer. But from the environmental point of view, the genetically engineered organism and try to use this for environmental applications, so that does not sounds very interesting at all. So then the idea here is actually, is it possible for us to identify certain environmental cues that can be used to boost CDJP level, and then we can integrate that into the operational kind of a process, and then we can also make the biofilm more robust. So in this specific case, we look at this bacteria, as what I mentioned just now, this bacteria was believed to be an aerobic organism and use oxygen as the electron acceptor. But when we look at the genome sequences and we compare the genome sequences with other organisms too, and we found this organism, in the genome sequence, in the genome, they also contains genes that are responsible for the nitrate respiration. So that means it is also able to use nitrate as the electron acceptor and convert it to nitrite. But the interesting thing is actually nobody demonstrated before. And we grew the biofilm of this organism, and we just leave it at the bulk aerobic condition. So that means in the media, in the growth media, we have oxygen in the media that's air saturated. At the same time, we add some nitrate in it, and we want to see what happens. And the interesting thing is that if you look at the nitrate concentration, nitrate starts to decrease after about 60 hours. So in the first 60 hours, you do not see much difference. At the same time, nitrite increases also after about 60 hours. And 60 hours sounds very important here. So then we look at the biofilm on the confocal, and this is what we have. So before 60 hours, we do not see much three-dimensional structures in there. And after 60 hours, we start to see the three-dimensional structure. So we believe that it is because of the oxygen limiting inside the biofilm. So then nitrate start to be used as the electron acceptor. So in this case, the nitrate can be reduced inside this biofilm under bulk aerobic condition. But the most interesting thing is actually if we look at the nitrate respiration by these bacteria, and we found the CDJP synthase genes are upregulated. Many of them are upregulated. And then we quantify CDJP level. And this is what we have. So if you use nitrate as the electron acceptor for this bacteria, and we have a much higher CDJP level in the cells. So this kind of interesting, like we are able to use the environmental cues to actually boost the CDJP level. Now the next question is, after we introduce nitrate to the growth media, whether the biofilm becomes more robust or not. So then we grow the biofilm with or without nitrate in the media. And this is what we have. With nitrate in the media, so the cells use oxygen first, of course, and after that, they will use nitrate as the electron acceptor. But, and you can see actually the cell viability is higher with the presence of nitrate in the media. And at the same time, so we introduced 3CA. You remember in the previous studies, we talked about 3CA. Once you introduced it, the biofilm start to disperse. Now in this case, we introduce 3CA in a condition where nitrate is introduced or not to the media. And this is what we have. With the presence of nitrate. With the presence of nitrate to the cell detachment, after we introduce 3CA, this is no nitrate. So without nitrate in the growth media, the biofilm looks like very susceptible to 3CA. So once you introduce 3CA, you see the detachment. The y-axis is the detachment rate. So you see the substantially increased detachment rate. But in the biofilms developed during that process, when you have nitrate introduced to the media, and this is what we have. So even after the introduction of the 3CA, the biofilm is still quite stable. And this means that the environmental cues may be harnessed to actually boost some of the biofilm-based bioprocesses. But this is just the examples. So just examples using common monas. Okay, so to summarize, today I shared some of our recent work on the understanding of the biofilm contaminant interaction, and I show how we can use the insights to improve the biofilm-based bioprocess, and how can we translate that into some biofilm biotechnology. So the environmental contaminants at the concentration that do not kill bacteria at all, but they may influence the biofilm dynamics. So in this specific case, so the influence is through the interfering with the CDJP signaling. And then environmental cues that are able to alter the CDJP level may be harnessed to modulate the biofilm contaminant interaction. So if you know what are the cues that are able to do that, and then when you operate the reactor, you are able to adjust different parameters. And then CDJP dynamics can be used to control the biofilm dynamics. That may help us to develop some novel environmental biofilm biotechnology, including the biofilm-based chemical production. So this is just some of our recent work on this. So I'd like to thank Chelsea for the continued support, especially our very supportive director, Stefan. And then we also like to thank the funding agencies. And with that, I'm very happy to take questions. Thank you.
Great, thanks for a really interesting presentation there. We've had a couple of questions come in. The first being, do you think that this system that you've created would also be applicable in other microbes?
Yes, so that's a kind of yes, interesting question. And we tested another organism in the wastewater system, and it worked. But for other organisms, we do not know, so we have to test it. And the interesting thing is actually for some organisms, the CDJP background is very high. If the CDJP background is very high and very noisy, even if you introduce the gene circuit in there, you may not be able to precisely control the dynamics. But for some organisms, the CDJP level, the background is not that high, then that works well. And at the same time, we have a student actually help us to try to reduce the noise level in the organism. So that's for not for the environmental applications, that's for the biochemical applications. Yeah.
Sure. Next question that's come in. Anshu has asked, at what stage in biofilm cycle the impact of the CDJP regulation impacts the most?
So for us, actually, we are applying this to the established biofilms. Yeah, because for us, eventually what you want is actually to control, especially for biochemical applications, you want to control the thickness of the biofilm. If the biofilm is very thick, you have a lot of unproductive biomass at the bottom. If your biofilm is too thin, the overall activity is low. So you always want to keep the biofilm at an optimal thickness. So for that, it's actually established the biofilm. We just want to remove the top layer or bring back the top layer only. Yeah.
And finally, in terms of implementation, what would be the key way that you personally would like to see this system being used for the benefit of Singapore, being that's where you are, and then potentially kind of externally as well?
So if you're talking about applications, I still think if we're talking about the applications for the light controllable biofilms, I think it may be used for like the biochemical production or the even biomedical related to research or biomedical related that area. But for the environmental, I still do think that the genetic manipulation is not the way to go, at least at this stage. But the environmental cues, explore that. I think that should be that may have some kind of interesting applications for environmental applications. Yeah.
That's great. That's all the questions. Thank you very much.
Thank you. Yeah, so if you have any other questions or if you want to know more about our research, feel free to send me an email or just contact us. Thank you.